Um, yeah, and I just wanted to let you know that I am on service today, so I'm gonna have to step away. So you will you can moderate your own questions, I am sure,
No
very
worries.
capably.
Happy to do
Okay.
that.
Okay, great, thank you. Anyway, I wanted to thank everyone for joining us. This is the Frontiers in Child Health Research Seminar, and as you know, we hold it on the second and fourth Mondays from September through June. Our series includes speakers with work focused on child health from both UCSF as well as guest speakers. And please let me know via email if you're interested in speaking or would like to suggest a speaker. Looking ahead, our next speaker will be on November 13th. It's Dr. Fernando Gonzalez from the Division of Neonatology. We're still virtual and using Zoom, and we ask that you stay muted unless you have a question, and Marina will open it up at the end to questions as well. Today, we're welcoming Dr. Marina Sirota from the Bakar Computational Health Sciences Institute here at UCSF. Marina's research focuses on applying computational integrative methods to disease diagnostics and therapeutics. Her primary interest is in leveraging and integrating different types of omics and clinical data to better understand pregnancy outcomes and the role of the immune system and tolerance in disease. Her work has been funded by the March of Dimes, the Burroughs Wellcome Fund, and the NIH. The title of her talk today is Leveraging Clinical and Molecular Data to Study Pregnancy Outcome in the Era of Precision Medicine.
Excellent.
That looks great.
Well,
Thanks.
thank you so much for inviting me, Roberta, as well as the kind introduction. So let me, can everybody see my slides in the correct view?
Yes, they look good.
Excellent. All right, so I'm very excited to tell you about our research today. And before I start, I always like to say why I'm excited to be a computational biologist. And one of those reasons is that there's tons of data that's publicly available or available to us through other sources that make it an exciting opportunity to start asking questions with data and computational approaches. So these are some examples of publicly available resources, such as the Cancer Genome Atlas, which is a collection of over 10,000 cancer samples that have been profiled with a variety of different molecular technologies. The Gene Expression Omnibus is a data repository of molecular data and has pretty much every transcriptomic study that has been published and even a lot of unpublished data as well. In addition, in the last 20 years, we've been really creative of ways that we've been able to use technology. So we've been able to profile the genome and the transcriptome for quite a while, but we can also look at an antibody repertoire. We can study epigenetics through methylation. We can look at proteomics and metabolomics, and we can look at the microbiome. And now, in the last few years, we can make many of these measurements on a single cell level. So that brings us to ask questions of how do we integrate these data and how do we ask questions about disease using these exciting technologies. And finally, the last part on the side of opportunity is electronic medical records or real world data. And this is basically all the information that gets captured when somebody goes to see a doctor. We have access to electronic medical record data at UCSF, but also across the whole UC system. And this is an effort that my mentor, Atul Butte, has been leading for the past 8 years at UCSF, and it's quite exciting. So the focus of my lab is to figure out how to use computation and use both molecular and clinical data to understand health and disease across the lifespan. And we work on a variety of different clinical areas. We look at pregnancy outcomes, such as preterm birth, and that will be the focus of my presentation today. We look at autoimmune diseases. We've studied cancer immunology and organ transplant. And then more recently have also gotten into trying to use these approaches to better understand aging, for instance, Alzheimer's disease. But across all of these, there are two themes that are consistent in terms of our interests. One of those is women's health more generally. A lot of the conditions that we study are more common in women than they are in men. And also the role of immunology and the role that the immune system plays in disease. We formulate computational questions to identify biomarkers or understand diagnostics a little bit better, to understand disease mechanism, and to impact therapeutics. And I'll give you some examples of each of those throughout the presentation. The focus of what we'll talk about today is pregnancy and preterm birth. Preterm birth, as of course many of you know, is defined as live birth before the 37th week of gestation. Worldwide, about 15 million babies are born preterm, and about 1 million of these babies die within the first month of life. In nearly half of the cases for preterm birth, there's no clear cause. While numerous factors have been implicated and associated with spontaneous preterm birth, this is a figure from a famous review by Roberto Romero, we still don't know. The phenotype is so heterogeneous that we still don't know what is going on in each case. So infection, vascular disorders, uterine overdistension have all been implicated in preterm birth, as well as cervical disease, stress, as well as the breakdown of maternal and fetal tolerance. So we have really taken a comprehensive approach to studying this problem. We work with a variety of different types of data, starting with looking at genetics of preterm birth, exploring the role that the environmental exposures play in pregnancy outcomes, and that we've done in close collaboration with Dr. Tracy Woodruff here at UCSF. We've looked at the microbiome, specifically vaginal microbiome, and which microbes might be associated with adverse pregnancy outcomes. And hopefully I'll have a chance to tell you a little bit about that work today. We've used transcriptomics, or gene expression, which genes are turned on and which genes are turned off. And I'll talk about that first. And finally, we have efforts to use electronic medical records data, both at UCSF and across the whole UC system, to better understand pregnancy and pregnancy outcomes. And I've been incredibly fortunate to have a number of very talented trainees throughout the years, and some of them are listed here, those leading the projects. So I'll start by talking about transcriptomics, but I guess I'll pause for a second. Are there any questions so far? All right, so I come from the background of genetics and gene expression, and we wanted to ask here, can we use gene expression data to identify new potential biomarkers for women who are at high risk for preterm birth? Previous studies a lot of them have had small sample sizes. Some have been reproduced, others have not. And a lot of placental samples have been used in studying transcriptomics in the context of pregnancy outcomes. And of course, those are not really accessible for diagnostics. So we started out by performing a cross-study meta-analysis to better understand the gene signatures in both moms and babies who are moms who end up delivering preterm and babies who were born preterm. But we start with the maternal signals. When we started this study, this is a few years ago, and the work was led by Bianca Vora, who was a very talented graduate student here at UCSF. And this was actually her rotation project in my lab. We identified at that time 3 publicly available, these are microarray data sets, so they were generated now nearly 10 years ago, looking at signals in maternal blood. You can see the sample size here, as well as the study design. It was different across these 3 studies. So one of the studies had about 75 cases and roughly equal size of controls. One of the studies had some temporal information, so samples from 2nd and 3rd trimester. And the 3rd study, in addition to maternal blood, which is here on the bottom, the sample size is quite small, but they profiled other tissues of interest, including cord blood. So the first thing that we need to do is figure out whether we can combine these data. And these are dimensionality reduction plots. They're PCAs, where each dot is an individual sample. And they're colored by which study they come from. You can see before the normalizations, the 3 studies cluster completely separately. However, we're able to overcome that by normalizing and carrying out batch correction. And that's shown on the 2nd graph here. Once we remove several outliers, the 3rd graph shows the same samples, but colored by outcome, where light purple is preterm and dark purple is term. And you can see that there's no clear separation between these groups. They're sort of intermixed. Maybe there's a slight shift. We've looked to make sure that the groups that we're comparing have roughly equal gestational age at sampling, and that is the case that's shown on this graph to the right. And in total, we have about 340 samples. This is looking at the signature for spontaneous preterm birth in these data sets. It consisted of about 200 genes that were significantly associated, significantly differentially expressed between cases and controls. This is a heat map where genes are on the y-axis and samples are on the x-axis. You can see that these two bars at the top, the first one shows outcome. Again, light purple is preterm and dark purple is term. And the second bar shows which study the samples came from. So the good news is, again, we're able to overcome the batch effect and the data does not cluster by study. But looking at this very top bar, I would say it doesn't really cluster that well by outcome either. There is this subcluster here that might be somewhat slightly enriched for women who deliver preterm, but overall, there's no clear separation, just like we saw in the overall dimensionality reduction. However, we do identify these 210 genes, 145 downregulated, 65 upregulated, and some of them individually could be potential biomarkers. We looked at network analysis and we saw that a lot of them were connected. Each node here is a gene or a protein, and they're connected together if there is some common functionality between them. When we looked at the pathway enrichment, we saw a lot of immune pathways misregulated. We saw that the innate or the non-specific immune system was upregulated in women who deliver preterm, and adaptive or specific immune response was downregulated. And these are some example pathways in the downregulated group, such as lymphocyte activation, T cell activation, and so forth. In terms of the individual genes and proteins, here are all out of those 210. These are ones that are secreted or can be found in measured by protein levels. And the two that are highlighted actually appear in the earlier time point for one of the studies. And that's IL-1 receptor 1 and TFPI. Both of these are quite interesting. Interleukin-1 receptor 1 encodes a cytokine receptor that belongs to the interleukin family. It's associated with several diseases, including autoimmune diseases and infection response. And IL-1 receptors 1 and 2 have been shown to appear in terms of protein abundance in pregnant rat uterus at term, and have been shown to be regulated by progesterone. So definitely a very interesting gene and protein to follow up. TFPI is tissue factor pathway inhibitor. It is involved in blood coagulation. It inhibits activated factor 10, and elevated plasma levels of this gene and protein have been associated with early onset preeclampsia and more generally associated with placental development. So if you recall, I mentioned that one of the data sets, in addition to maternal blood, also explored fetal transcriptomics through cord blood profiling. So it's a much smaller sample size, but here you can really appreciate the clustering and the separation between the infants that were born preterm versus babies who were born at term. There's a larger gene signature, almost 500 genes. But the interesting part is that the pathway signals are reversed between the mom and the baby. Of course, we don't know here whether the difference that we observe is anything related to preterm birth or the overall immune development of these babies. But there is some sort of maybe some sort of an interplay between the maternal and the fetal immune system. Because for all of the genes that are significantly associated with preterm birth in both moms and babies, the signal is inverse, which means that a gene that is upregulated in moms who deliver at term is downregulated in babies who are delivered at term. And this is true across the board for all of these genes. There are some interesting ones here. TLR5, for instance, plays a fundamental role in pathogen recognition and activation of innate immune responses. It's been associated with inflammatory, it's the activation of this receptor mobilizes NF-kappa B, which in turn activates host and inflammatory-related target genes. TLRs more generally have been shown to be involved in pro-inflammatory and pro-labor mediators response. And then the variants in these genes have been associated with both preterm birth and bronchopulmonary dysplasia. So again, a very interesting potential candidate to follow up experimentally. So overall, I've shown you how we've taken publicly available data, and given these are data sets that have been around for a while, we use them to ask some new questions, both about the maternal transcriptomic signals in preterm birth, and as well as the relationship between the immune system of the mom and the baby. As a follow-up, I don't have time to get into the details today, but we've launched a study in collaboration with Dr. Tippy McKenzie to look at the immune repertoire, both in maternal and cord blood at delivery, to better understand this interplay. But what I wanted to tell you about next is ways that we were able to use this signature for drug discovery. And I'll pause for a second here in case there are any questions. All right. So in terms of drug discovery, a method that we've been developing in the lab, an approach that we've been using, is drug repurposing. The idea is it is very expensive and it takes a long time to take a drug and bring it to market. And 90% of drugs fail in early development. A solution to that is to take a drug that's already FDA approved and try to find new uses for those drugs. Obviously, it reduces the risk of failure because many of the compounds have already been tested and have gone through safety testing, and also, as a result, lowers the cost of discovery. Of course, we need to be understanding of the potential toxicities and the fact that these tolerated toxicity might be different depending on the indication itself. So for instance, the type of toxicity that somebody might be willing to tolerate for the sake in the context of cancer might be very, very different than for a chronic condition like type 2 diabetes. And of course, this is way different when we start looking at vulnerable populations, for instance, pregnant women. So in my lab, we have developed a pipeline for computational drug repurposing based on gene expression data. And the idea is, given a disease signature, which is a set of up and down regulated genes for a certain disease, we can query it against a data repository of gene expression data on drugs and potentially identify drugs which reverse that gene expression signature back to normal. So the idea is, if the disease signature and the drug signature are opposite of each other, then maybe that drug could be used as a therapeutic. This approach was developed when I was actually a graduate student with Atul Butte, and since then we've applied it to a variety of different conditions, starting with IBD, dermatomyositis. Cancer is a natural application because the expression of the drugs is actually measured in cancer cell lines. And of course, during the pandemic, we looked at COVID-19. But today, I'd like to tell you about our work, drug repurposing efforts in the context of preterm birth. So I told you about the signature that we generated, and we wanted to see if we can use it to identify therapeutics to modulate preterm birth. We used the set of 210 genes that I mentioned before on the disease signature side. On the drug side, we used the resource called the connectivity map. So this is a data set that was originally developed by the Broad Institute, where they've taken about 1,300 drugs, put them on various cancer cell lines, and measured expression before and after treatment. Again, the caveat is this is cancer cell lines. There might be certain genes that are not even expressed, certain genes of interest that are not even expressed on the cancer cell lines themselves, but we're looking at the differential. So this is a genome-wide study, and they had over 6,000 experiments, so different dosages, different cancer cell lines across these 1,300 compounds. There's another effort in terms of drug-related data generation that's worth noting, and that's the LINKS data set. Here, they looked at tens of thousands of compounds, but instead of genome-wide, they only measure 1,000 landmark genes, a little bit under 1,000, with the idea that the rest of the genome expression can be imputed. And I'll be honest, in our hands, that imputation was not very effective. So we stick to the genome-wide data set that I mentioned first, the connectivity map. So I know FDA pregnancy categories are no longer used actively, but we needed some way to filter our results. So we still focus on category A and B drugs. In terms of the results, we found 83 compounds that significantly reversed the preterm birth expression signature, and 13 of them fell into the pregnancy A and B categories. Four have been previously investigated for preterm birth, either through animal or epidemiological studies. And this work was led by Brian Lee, who was a very talented postdoc in my lab and published in JCI Insight in 2020. So these are the 13 drugs. Here on the right-hand side, you can see the spontaneous preterm birth expression signature, and you can see that these compounds reverse it. One interesting one to note is progesterone. It is the furthest down here, and it was the only FDA-approved drug for preterm birth. It was not effective, not that effective, maybe in only 30% of the women, and it's no longer widely used, but nonetheless, it gave us some inkling that we might be doing something right. Others that have been explored in the context of preterm birth include metformin, folic acid, and clotrimazole. However, here on the left-hand side, you can see a network. Each pink rectangle is a compound. Each blue oval is a target of that compound. And you can see that right next to progesterone, there's another drug that shares a number of targets, yet nonetheless is different. And that drug is lansoprazole. Lansoprazole shares several targets with progesterone, including CYP1B1. So we thought that it might be interesting to explore a little bit further. Lansoprazole is over-the-counter proton pump inhibitor. It has been shown to induce the stress response protein heme oxygenase 1, and inducing the expression of HO1 has been shown to reduce pregnancy loss in HO1 deficient mice. Our hypothesis was that by targeting heme oxygenase 1 expression, may have potential in preventing recurrent pregnancy loss. So we collaborated with David Stevenson at Stanford and his team, who have had an interest in heme oxygenase 1 for many, many years, to launch a pilot validation study in a LPS-induced inflammation fetal wastage model. And of course, I recognize well, first of all, I will say I'm not an animal model expert by any means. I'm a computational person. And I recognize that pregnancy in animals is very, very different than human pregnancy. Nonetheless, this was a starting point. So in this experiment, pregnancy was confirmed on day 0.5. The model was induced as in day 7.5, and outcomes were explored at day 12.5 of pregnancy. We carried out treatment with both progesterone and lansoprazole separately on days 6, 7, and 8. And all this work was done in collaboration with Ron Wong and Sore Iwatani and David's team. And finally, we looked, as I mentioned, at the number of viable fetuses as an outcome. So here is just the disease model. On the right-hand side are just healthy animals. LPS 100 is a disease model that got induced, and you can see there's quite a bit of heterogeneity. Nonetheless, there's a significant decrease in the number of viable fetuses. And this is the same model, but in combination with oil and DMSO, because those are the solutions that the drugs were delivered with. When you look at the drug treatment, again, this is the animal model. This is the treatment with progesterone. And on the right-hand side in purple, you can see treatment with lansoprazole. You can see a significant return back to normal with lansoprazole treatment, which was not observed with progesterone, at least based on this study. We have since collaborated with Sing Sing Wei in Cincinnati to explore this further. He has developed an immune-based model of preterm labor with the use of FOXP3 CD4 positive T cells. The way that they've done it is they deplete these Tregs, which they have shown causes fetal wastage and loss of live pups. They've shown that this fetal wastage is associated with expansion and activation of maternal CD8 positive T cells. And these results are shown here, and they have been published previously. So what they have done is they have shown that exogenous progesterone protects against fetal wastage through partial transient maternal FOXP3 cell depletion. And that's shown here. This data was their own data published in iScience in 2022. So this is showing percent of fetal wastage, as well as the number of live pups, as well as the molecular frequencies of the actual cells and spleens in the spleen of this endocyte of these animals. So you can see a significant reduction in fetal wastage with progesterone treatment and a higher number of live pups. This experiment since has been repeated with lansoprazole treatment, and these are very preliminary results here. In addition to lansoprazole, we also looked at another drug in a similar family called famotidine, and we could see similar results in terms of, obviously increased fetal wastage with the disease model and a decrease with lansoprazole treatment, a partial rescue with famotidine. This is the number of live pups, and you can see it significantly go down with the disease model, again, getting rescued with lansoprazole and partially rescued with famotidine. These are the cellular assays as well. So this is incredibly exciting to us. Again, mouse pregnancy is very different from human pregnancy, but nonetheless, it demonstrates that we could use publicly available data to make these predictions and then see at least some alleviation of the phenotype in 2 independent animal models. We're currently collaborating with Brice Gonier at Stanford and his team, who have developed a multiplexed mass tag barcoding drug screen to look at what these drugs do in actual relevant human cells. I mentioned that the data that we use for prediction is in cancer cell lines. That's not great and not always applicable to the actual conditions that we look at. So this is a way to look at what these drugs do in actual human cells, in PBMCs, let's say of even pregnant women. These are very preliminary. The font is not showing up, but you will have to trust me. These are very preliminary results on lansoprazole in PBMCs, and we've seen activation of JAK-STAT response in CD4 positive T cells and inhibition in MyD88 response in another cell type of interest. So again, I've shown how we use expression data to better understand what is going on the transcriptional level in moms who end up delivering preterm. We predicted and validated lansoprazole in several mouse models, and we're collaborating to better understand what these drugs do in relevant human cells using mass spectrometry. Also in the lab, we're expanding this approach to other conditions of interest, such as preeclampsia and endometriosis. So I'll pause there for a minute before moving on to the rest of the stories. Any questions?
Yeah, Marina, it's Mel.
Hi, how are you?
I'm good. How are you? Enjoy this.
Doing
Just curious, lansoprazole, as you know, as everybody knows, PPI, famotidine is an H2 receptor, different mechanism, and yet are you saying that the data show that they go after the same immune functions, and that's why you tried the 2 different drugs?
No, well, they're both used, as I understand, for GERD. So that's why we wanted to look at them, because they have different mechanisms. So the experiments that we've done so far tell us nothing about mechanism of action. They just, so the prediction part, we can look a little bit at the pathways, right? But only we've done the prediction for lansoprazole. For famotidine, we just wanted to see if, what is the question was, what is the alleviation of the phenotype related to? Is it the target itself, or is it something different about what the drug does? And I'll say those results are still very, very preliminary. Basically, we see a smaller reduction with famotidine. And actually, the data is so preliminary, I don't even know if it's statistically significant yet. The lansoprazole results seem to be. So we're still exploring that a little bit further.
Thanks.
Sure.
Hi, Marina. I'm Betsy
Hi.
Crouch. I've heard a lot about your model, so it's great to hear you speak. I've noticed that you're taking samples from maternal blood, so you're getting a lot of immune cell hits, which makes sense. Do you think that you're going to be able to pick up hits from other cell types, or have you seen any of interesting candidates in that regard?
So great question. We did do, so these are all bulk data sets that I'm talking about today. Of course, now we have single cell data. We have not done the drug repurposing study for single cell data in the context of preterm birth. We are doing that for Alzheimer's disease and for endometriosis to basically with the goal of coming up with combination therapy that targets specific cell types or signatures in specific cell types. And in Alzheimer's, we actually have some really exciting preliminary data for combination that seems to be working quite well. The answer is yes, we need to do it. We haven't done it here. What we have done that I didn't show is in bulk measures, you can run cell type deconvolution and cell type enrichment analyses, which allow you to quantify the amount of different or relative amount of different cell types. This is all predicted in a sample of interest. And we have done this for the first study that I mentioned. And it basically corroborates the innate and adaptive kind of split that we saw on the maternal side in terms of upregulation and downregulation. So we've done that, but we haven't related it to the drug data, which is something that we're doing in other diseases.
Yeah, very cool. I think so. I'm a vascular biologist, so I've always been interested, in the contribution potentially of the vascular, the placental vasculature, but in general, the vascular, let's say like maybe role of the vascular cells, but also, there are consequences of vascular cells. We know, for instance, on babies who are born IUGR. Can you pick up any like transcriptomic signature from the vasculature when you're looking at a blood sample? Do you know?
That is a good question. So with the deconvolution, we did not see that, but on a single cell level, maybe. I mean, we can also, with a specific question like that, you can query the data for certain pathways that are involved in vasculature and see if they come up. Maybe we won't see much if we look genome-wide, but if you have a specific hypothesis, then that's something that could be explored.
Cool. Yeah, thanks for letting me ask about my favorite cell type.
Sure. And I can look it up and see. I think single cell data will be more informative, and I think there are some data sets in the context of preterm birth now. We just haven't gotten there yet. Yeah,
we could take it offline, but yeah, and I think there is potentially interesting interactions between the vascular and the immune cells,
Absolutely. And there are approaches to look at that specifically in single cell data using methods like cell chat, looking at cell communication and how they're correlated. It's very cool. We just haven't done it in this context yet.
Cool, thanks.
Sure. Other questions? All right, well, I'll shift gears and tell you a little bit about some of our other efforts. So we started with the study using publicly available data, and it really allowed us to see what kind of questions we can ask. Around the same time, I was approached by the March of Dimes to create a data repository for preterm birth. And this is something that we've been now involved with for almost 10 years. So we created this resource, and it's co-led by myself and Dr. Tomiko Oskotsky, who's a senior research scientist in my lab, with the idea that this will be or is a central data repository for omics data across all the 6 prematurity research centers funded by the March of Dimes. The database is rich. It has transcriptomics, genetics, microbiome, proteomics, cytoph, methylation. And the whole idea is to enable data integration and enable new scientific questions, enhancing collaboration across these centers, but also beyond to the general research community. The database was launched as a separate launched in 2017, but launched as a separate independent resource in 2020. It's regularly updated. This is the link if you want to take a look. It currently has 64 studies, almost 30,000 participants, and 41,000 experimental samples. We track the downloads, and we can see that people are using it pretty extensively. It's searchable by assay type, biosample type, which center it came from, as well as different conditions. So preterm birth and preeclampsia are widely represented, obviously. We've also been using this data for education. One of the efforts that I run is called AI for All. It's a high school program. It's a 3-week high school program over the summer. And again, I co-run this effort with Dr. Oskotsky. And the idea is to introduce high school kids to applications of artificial intelligence in biomedicine. So for instance, one of the projects relating to women's health from the past 2 years was predicting reproductive health outcomes using microbiome data. And students have looked at both preterm birth as well as recurrent pregnancy loss. Another effort to get other researchers, especially researchers from other fields, to use these data and to study the question of preterm birth that we've applied is crowdsourcing through these dream challenges. So a dream challenge is a crowdsourcing challenge which examines a certain question in biology and medicine. And the goal is to bring a computational community, so even computer scientists or statisticians who don't do biomedical research at all, to the data and to the questions of interest. And the question that we were interested in asking was, how can we predict women who are at a higher risk for preterm birth? And here we wanted to use microbiome data. We've had a previous study, a meta-analysis. This was led by Edith Costi, who was a very talented postdoc in my lab. And the work with her demonstrated that women who deliver preterm tend to have a more diverse microbiome, especially early in pregnancy. That's shown here on the right-hand side. You can see the orange line is a modeled variance of women, of the microbial diversity of women who deliver preterm as opposed to women who deliver at term. And this is across a few hundred samples. We could also pinpoint the specific bacteria that are associated. Some of these have been associated previously, like Prevotella and Gardnerella. Lactobacillus is inversely associated, as shown by the kind of light purple color here on this heat map. But then we were able to find some new associations as well. The growth of microbiome data has been exponential in the past 10 or 15 years. This is a graph from a paper by Katie Pollard, and you can see that really there's tons of microbiome data that's publicly available. And we got really interested in integrating it. Well, we learned it's not that easy. There are many different biases. There is 60ness versus whole genome. There is taxonomical versus gene level analysis. Obviously, sample prep is often different. Even within 60ness, different regions of the variable genome, or different variable regions of the 60ness gene are sometimes captured. And of course, study design is often different, longitudinal versus case control. So we went to our own data repository and supplemented it with data that is publicly available beyond the March of Dimes funded centers and identified these 9 studies. You can see the data here. Where does the study come from, number of samples, as well as which region of the 16S gene was profiled. And the question was, can we predict women who are at high risk for preterm birth? You can see that it truly takes a village to put something like this together. We aggregated, the effort was co-led by Tomiko Skotsky and Jonathan Golub, who's one of our collaborators from Michigan. And again, many groups involved, Sage Bio Networks. Obviously, this was all funded by the March of Dimes, as well as a number of other institutions. So this is the data that we had. 9 studies, almost 1,300 participants, 3,500 samples. And we had 2 tasks: predict women who are at risk for preterm birth and predict women who are at risk for early preterm birth. But the first task is to aggregate all this data. How do we combine that? There is an approach that one of our collaborators, Jonathan Golub, has developed, and it works to aggregate microbiome data very effectively. It uses phylogenetic placement of the actual sequences. So basically, it creates a phylogenetic tree of the sequences once you load all the sequences in, and it really allows us to get all these studies on the same level. The approach is available as a preprint and as a package, and the paper actually just got accepted for publication. This is what the data looks like before and after processing with Maliampi. Each dot here is a sample. You can see that they really cluster by study here on the left. And these are the 9 studies, as well as 2 independent validation datasets that we generated with collaborators. You can see that after the normalization, there is still some study-specific effect, but we were able to get all this data on the same plane. And this, for microbiome, this is a huge deal. For expression and genetics, we know how to do this well, but for microbiome, it's very hard because of all the heterogeneity. But nonetheless, when we plot the same dots, but colored by outcome, we can't separate term versus preterm very clearly. And you can see that here, where individuals who deliver a term are shown in blue, and individuals who deliver preterm are shown in orange. And this is really where machine learning comes into play. So we launched this challenge. It started in June 2022. The teams were from all over the world. We had 318 teams register and over 100 submissions for each of the 2 questions that I mentioned: prediction of preterm birth and prediction of early preterm birth. The teams had a few months to develop their models, and then we evaluated everything, and this was the results were presented last November at the DREAM conference. The models were assessed using ROC curves, so this is fairly standard, and again, we had 2 tasks. And as I mentioned, we had 2 independent data sets, one from Wayne State and one from Stanford University. This is data that none of the participants in these competitions have seen before. So we basically aggregated all this data. We made it available to computational researchers across the world, and we had them submit their models for evaluation on the data that they have never seen before. And this is what the results look like. For task 1, which is prediction of preterm birth, we got accuracy of almost 0.7. And for prediction of early preterm birth, we had top results of 0.87, which is actually pretty good. We used a number of different approaches to evaluate these, using bootstrapping, oversampling, undersampling. Basically, we tried to break these results to the best of our ability and make sure that they're robust. And these are the winning teams. Everybody presented their findings in Las Vegas about a year ago, and this paper is under review in Cell Reports Medicine. So this is an example of a way that we can take publicly available data, we can aggregate it, make it available to the computational community at large, and ask them to build computational models. What we're doing now is we're trying to better understand the features that came out of those models and see if we can maybe modulate them or use them as biomarkers. So our goals are to better understand what is underlying the predictions that we've observed, and the predictions are fairly good. Now, the question is, how can we combine these data with other data sets as well? To maybe by integrating transcriptomics and microbiome, we could get to the answer a little bit better. So in the last few minutes, I want to talk a little bit about our work leveraging electronic medical records. This started as actually a high school student project a summer project in my lab with a very talented student, Carolyn Wang, who is now in Columbia, or maybe already graduated from Columbia, under the mentorship of Edith Costi. And we thought electronic medical records would specifically be amenable to looking at pregnancy because we have the outcomes information. We know when these women delivered. The period is roughly 9 months, and there's some consistency in terms of the clinical care for pregnancy. So we can actually build trajectories. At that time, we had data on about 15,000 deliveries, again, demographics, lab test results, diagnoses, codes, and medications, and we wanted to see if we can identify those at higher risk for preterm birth. So we built a trajectory of pregnancy, going backwards from the day of delivery to all the diagnoses during pregnancy, as well as medical history, demographics. So we have medication information as well as lab test results, in addition to ICD codes for all of these patients. Data is messy. Many of you know that way better than I do because you actually are heavily involved in aggregating and inputting a lot of this information. We often have, and you know how messy the electronic medical record can be. Nonetheless, it's an opportunity. So data cleaning is a huge step of what we do. By the time we clean the data, we cut our sample size here by half. Nonetheless, we could identify some of the known relationships. For instance, we could see that women who are Black or African American have a higher rate of preterm birth in our data. We could see for specific associations, prior preterm birth is the biggest risk factor. We saw that obesity, type 2 diabetes, and ancestral hypertension were all associated. But we could also see things like anxiety disorder, major depressive disorder, and hyperthyroidism, which are a bit less studied and worth exploring. We have further collaborated with Tony Capra while he was still at Vanderbilt to build predictive models for preterm birth. They actually started at Vanderbilt with a cohort of pregnant women and build machine learning models to predict which women are at a higher risk for preterm birth. Then those models were sent to UCSF. So these are the patients at UCSF. These are the patients at Vanderbilt. And we could see very similar performance across the board. You can see that AUC is 0.7 or 0.8, so not amazing, but nonetheless, there's predictive value. But the more impressive part is that they're similar across the two institutions, even though all the training here was done at Vanderbilt. So right now at UCSF, we have data on about 50,000 deliveries across over 30,000 moms. We recognize that this data might not be fully representative because we're a tertiary care center. So we have the preterm birth rate is actually 16%. Because we have curated outcomes information from the OBGYN department, we actually know which women deliver preterm spontaneously as opposed to medically indicated. Across the whole UC system, we have data on about 140,000 individuals, which is quite impressive. And for everybody, we have measurements such as drug exposures and other clinical conditions. These are just the demographics across the UC-wide cohort. And we have a number of different projects. We're continuing to apply predictive modeling and looking at subtyping for both preterm birth and recurrent pregnancy loss. We're looking at drug exposures in pregnancy. Specifically, there's a study that we have in collaboration with the K Institute where we've looked at SSRIs, but we can extend that to others. We're studying clinical notes to better understand clinician bias. And of course, we're applying these approaches to other conditions of interest. In one of our collaborative studies, we have went beyond just the maternal data. We can link the maternal and the neonatal data to try and predict neonatal outcomes using maternal electronic medical records. This is a study that was led by Nima Agapur and his team at Stanford, where they developed machine learning models to predict neonatal outcomes. They sent those models over to us at UCSF, and we were able to replicate by matching the ICD codes by frequency first, and then applying these models, we were able to achieve very similar results across the two institutions. So this, I'm not going to get into details in the interest of time, but this just shows similar performance across Stanford, shown, I believe, in red, and UCSF in blue. So my hope is that I gave you some idea of ways that we use both clinical and molecular data to ask new questions, to generate hypotheses. Of course, many of these need to be followed up with experiments in the lab and or further clinical trials, but at least it's a way to get started using data and computation. With that, I'd like to thank everybody on my team, as well as collaborators across both at UCSF and across the March of Dimes network. The DREAM effort was really a huge number of individuals who participated, and all the database development for data sharing is done by the Peraton team. Of course, thank you for all the funders who support our work. This is my team in February of 2020, where we were all in one elevator. Because our work is computational, we've been doing a lot of remote work, but nonetheless, this is the group. And I would be happy to take any questions in the last few minutes.
Thanks very much, Marina. I'm Tom Shimataki. Hi, I've seen your name. I've heard about you, and Yao Sun actually referred me to your work, and I'm sorry I couldn't join at the beginning of this talk.
No worries. I think it's recorded, but I'm also happy to chat offline.
Yeah. No, we also had Ivana Marek come and speak to our group of neonatologists last spring about the work that she was doing with preeclampsia prediction, I guess.
I've known Ivana for many years. We have so many papers together.
Yeah, it's wonderful work.
They do a lot of model building on their side, and they send stuff over to us, and we do a lot of model building here, and we send it to them to validate. So we work very closely. Wonderful.
Yeah. Obviously, it has a lot of bearing for our specialty in neonatology if we can predict perhaps more accurate predictions of preterm birth and which pregnancies are at risk for that. Have you had, has any of your work translated to the neonatal side? Have you looked at any predictions on the newborns in the NICU?
So the only thing is we have two projects, and both of them were not initiated at UCSF. We've just served as a validation for Nima Agapur and his team.
Yeah, so, Nima, I know.
So one of the projects was published in Science Translational Medicine. I didn't get into details because, again, they were the leads on that. But they built machine learning models to be able to predict neonatal outcomes using maternal data. So we can link maternal and baby data at UCSF, and we took their model. When we first ran it, it actually did not work. So we realized that there are differences across the two systems. And what we did is then we took the frequencies of ICD codes, and because things get coded differently in different institutions. So what we did, once we matched just by the frequency without any feature selection, we just matched by the frequency. And then we used those that were similar in frequency, were used at an equal frequency across the two institutions. Then you could see that the model worked very comparably. Nice. And the other project that we're helping them in terms of validation is looking at TPN orders. And that data, I will say, and I would love to chat more. We finally got it, but it's literally has taken 9 months, I think, to get access to TPN data at UCSF, because we've been figuring out how exactly it's captured, because they have some models and predictive analysis that they've done, associations with adverse outcomes based on TPN regimens. And we wanted to see if they're observed here. From preliminary analysis, it does look like it holds up, but we literally got the data last week. So yeah, so those are the two that I've been involved with, but we do have the matched data. So that if there are interesting questions that you would like explored, we can set up a collaboration and see. I'm not a clinician, so I'm excited to see, questions and learn what are the interesting questions in the field so that we can help.
Yeah, no, I think it's certainly the future, and I'm very curious to see how we can apply it to our, because we collect a tremendous amount of data, right? From labs to monitoring to other pieces of information that obviously we would love to see if we can make predictions from it. So thank you for your work.
Sure. Thank you so much. All right, I think we're at time. It was my pleasure to present, and thank you again.